All right, so here it is, the last part of chapter number 6. And well, this is kind of the last thing that we're going to talk about with respect to protein structure. And if you remember the central dogma of molecular biology, it's transcription, DNA to RNA, and then translation, RNA to proteins. So post-translational modifications. So these are modifications to a protein that, when do they take place? Post-translation. So when a protein has been synthesized, chemistry still takes place on those proteins. So these are known as post-translational modifications. There are a number of different post-translational modifications that take place in the cytoplasm as well as in the nucleus. Now, what happens is a protein gets synthesized at the ribosome, and then an enzyme does something to it. One of those modifications would be throw a phosphate group on there, throw a carbohydrate, throw a ubiquitin subunit on there, throw what's known as a sumo group. You know what, go ahead and acetylate that protein, or let's go ahead and methylate it. These are all different options of groups that can be added to a protein, and you can have a dramatically different outcome. Okay, so, or maybe not dramatically different, but a protein is going to be different if it's phosphorylated or if it's been ubiquitinated. We already talked about ubiquitin is generally a target for degradation. Now, phosphorylation of proteins is generally going to focus on a couple of different amino acids. So this would be right here, our serine residue, but instead of that OH, we've got OPO3. So that hydroxyl group is removed and a phosphate group is added. And that molecule is now known as phosphoserine. Phosphothreonine, that hydroxide group on a threonine residue is replaced with an OPO3. And my head's kind of obstructing it, so I'll just go ahead and draw it out here. Rather than an OH, an OPO3 group there is added to that phosphotyrosine. So those are pretty much your most common targets of phosphorylation. Now, how does that phosphorylation take place? Well, it needs a donor, something like adenosine triphosphate. So you can give up that phosphate group, form phosphoserine, and then have adenosine diphosphate left over, an ADP molecule left over. Now, phosphorylation, dephosphorylation of an enzyme can change the activity of that enzyme. You can kind of think about it as a little bit of a light switch, but it's not completely linear. So don't think about it as, well, phosphorylation equals an enzyme is activated. And likewise, don't think about it as phosphorylation equals deactivation of an enzyme. It's very complicated, or it's very variable. In some cases, phosphorylation might mean that an enzyme is turned on. In some cases, it might mean that it's turned off. But the addition of that is going to have an impact on the function of that enzyme. And you might think, well, why? Reason for that is because that phosphoryl group has a pKa of about 6.7. In addition to that, you're replacing, think about it like from a spatial standpoint, you're replacing this with OPO3. So you've got this group right here, an OH. And if you want to think about it in terms of like angstroms, an OPO3 is considerably larger than a hydroxyl group, in which case it's going to occupy a different amount of space. And therefore, the interactions that were present between an OH and something else are going to be different from the interactions between an OPO3 and anything in the surrounding area. The predominant species of a phosphoryl group is dianionic. Has 2 negative charges at a physiological pH. So now all of a sudden, not only is it larger, where a hydroxide group is going to be really good at hydrogen bonding, but a phosphate group now has 2 negative charges. So what's that going to attract? Could it attract some cations? The -2 charge likes to interact with arginine residues, and adding this phosphoryl group can make it change the conformation of the structure of the protein. So it might push some things out of the way. Now, like we said previously, it can turn on or off a function of a protein or lead to increased or decreased interactions with other proteins. So an example of phosphorylation cascade and metabolism, well, the adrenaline-driven cascade. So you've got a receptor protein that is associated with a complex of proteins. Now that complex of proteins is going to activate, so when you have your stimulus, so adrenaline stimulates the receptor, that receptor is going to activate a protein complex that activates an enzyme known as adenylate cyclase that takes ATP and makes a secondary messenger known as cyclic AMP. Now that cyclic AMP is going to serve as an activator of a protein kinase A, or PKA, and ultimately, long story short, what this is going to do is it's going to lead to the activation of an enzyme known as phosphorylase A. Phosphorylase A breaks, in human bodies, the sugar storage molecule glycogen down into glucose subunits. So through this series of steps, adrenaline leads to phosphorylation of an enzyme that leads to the release of glucose into the bloodstream. Kinases are the enzymes that actually carry out those phosphorylation events. Kinases attach phosphate groups onto different substrates. So there are kinases that are known as sugar kinases, in which case they add a phosphate group onto a sugar, but there are also enzymes known as protein kinases that add a phosphate group onto a protein. So this is relevant, but it's a little bit of an aside here. So the transfer of phosphates is not exclusively onto a serine residue. We'll see it when we get into the metabolism portion of this class and we talk about glycolysis. At any rate, that's the end of chapter number 6. I hope you have a good day.